Assalamualaikum and good morning to Puan Munirah Mihat. My name is Adil bin Muhammad Anwar. My matric number is 23DBT20F1016. So now I'm going to talk about our lab assessment, that is protein isolation simulation. So a little bit introduction about protein. So proteins are normally performing their function in a dense mixture of other proteins, carbohydrates, lipids, nucleic acids, salts, and other metabolites. They may exist free in solution as components of protein complex, absorbed to cellular structures, or as integral parts of cellular components such as membranes. So in the protein simulation, we will purify protein by column chromatography. So this protein purification simulation allows users to explore how a protein of interest, red fluorescent protein, can be purified from cell lysate using hydrophobic column chromatography. Column chromatography is preparative technique used to purify compounds depending on their polarity of hydrophobicity. So at the simulation, first I have been given an aliquot culture containing RFP producing E. coli cells. So my goal is to extract and purify the red fluorescent protein. First, I need to lyse the cells using lysis buffer, which breaks open the bacterial cell wall. This releases many different proteins, nucleic acids, cell wall components, and other cellular debris. Next, I will use centrifugation to isolate the soluble components at the cell, which include the protein of interest. And finally, the protein in the solution will be subjected to column chromatography, which specifically isolates and purifies the desired RFP protein. So, effect of each chemical to the molecules or cells in hand. It is important to remember that I am working with biohazardous bacteria, which is E. coli. This means I should use aseptic techniques and work in a sterile environment to prevent contamination. I should not touch anything that had been in contact with the bacteria directly, and must only hold equipment on the outside if necessary. There should be a strict disposal of all used tubes and pipette tips into a biohazardous waste container.
So now I'm going to do a simulation about purifying protein by column chromatography. So repeat the same step for 3 times.
So after the simulation that I have done, the result is during the process of protein purification, different proteins will travel at different speeds in the column, allowing me to isolate and collect the protein of interest. Since the red RFP is very hydrophobic, it will bind more tightly to the bits and travel slowly through the column and be one of the last proteins to elute. So in the picture, we can see that ideal cell culture results. There will be red and slightly cloudy. The bacteria replicated in LB, AMP or ARA and produce RFP which remain inside the cells. So that's all from me. Thank you.